and welcome to Biology Practical 4, Osmosis. Now, osmosis is the movement of water from a high concentration to a low concentration through a semi-permeable membrane. Now, we test this in quite a simple practical, which is shown here. Now, what we do first is we get our potato and we bore a simple hole through and remove the potato core. Now, these should roughly be the same size for everyone to try to make the test fairer. Now, after we have done that, trimmed it down, okay, we weigh it to get our mass before, okay? So for example, this one will be 0.223, all right? After we've done this, as I've set these up before, we have our solutions. 0%, which is pure water, 1% salt solution, 2, 3, and so on, okay? We put our weighed potato into the solution, like so, and wait about 20 to 30 minutes for this to occur. After we've done this, we remove the potato, blot it dry with paper to make sure our results are fair and no water is being lagged, okay? And we weigh it again to get our mass after. Now, Mr. Taki will now go over the results section for you.
Right, after you've done the experiment accurately, you come out with a result that looks like this. So you have a table where you have the concentrations from 0, which is pure water, to a higher concentration. You can see that the masses at the beginning and the end, the masses at the beginning don't really matter because we are actually looking at the change in mass. So for the first one, for example, we had a mass of 5 at the beginning, 5 grams. And at the end, you had a mass of 8. So the change in mass is 8 take away 5, which will give us 3. And then the second one, you had a mass of 4 at the beginning and a mass of 6, which gives us a change in mass of 2. For a concentration of 2, we had a mass at the beginning of 5, a mass at the end of 5, which shows us no change in mass. And as you go down, the higher the concentration, you realize that the changes in mass, instead of the mass increasing here, the mass has actually decreased. You can now use this result to calculate your percentage change. It is better to use percentage change because you can compare, it makes it easier for us to compare the results. Now, I've just shown you how to work out the first one. To calculate the percentage change, this is something that is very common in biology questions, in biology papers. You need to know how to do that, which is you look at the final result, take away the initial result over the initial result times 100. So to get 60, I go 8 take away 5 over 5 times 100, and that gives me 60%. They might ask you to plot this on a graph, or they might give you a graph where you need to interpret. If you plot this on a graph, the graph will look something like that. So you have concentration on this axis because that is the concentration on that axis because that's what you change, and then the percentage change in mass in that axis. Your graph will look something like this. So you go across, and that's your x-axis, and that is your y-axis. Where the graph crosses the x-axis tells you what the concentration of the potato is, because there was no change in mass at that point. You can also see it on the table. On this table, for example, we can say the concentration of this potato is actually 2, or the liquid is actually 2. Because if you compare the concentration of the potato and the concentration of the liquid, there was no the net movements here were the same because the concentration on both sides were the same. That is why the change in mass was 0. So very common, you will have a graph, you need to interpret, you might have a table. One thing with graphs as well is to interpret a graph properly, you need to make sure you know what each small square represents. On the x-axis, what does each small square represent? On the y-axis, what does each small square represent? If I have, for example, let's say 1, 2, 3, 4, and I go from 0 to 4, I know that each small square here represents 1. So that is 1, 2, 3, and 4. And if I'm going from 0 to 20 up there, then each small square represents 5. So you should be able to, if you know how to interpret graphs and know how to interpret results on table, then you're going to do well in this experiment. We hope this video helps you to achieve the best grade in the osmosis experiment. And remember, they can ask you to describe the process of the experiment, or they can ask you to interpret the results from the experiment. We wish you good luck.